Hello everyone, and welcome to this month's webinar on trial design issues for Phase 2 trials. Phase 2 is usually the first opportunity to test if a candidate therapeutic has a positive clinical response in humans, and is also an opportunity to gain insight into the optimal dose for the proposed drug to be brought forward into a Phase 3 clinical trial. However, Phase 2 is also the stage of the clinical trial process with the highest failure rate, so good study design is crucial to make informed decisions in a timely manner. In this webinar, we will cover the challenges faced when designing Phase 2 clinical trials, and we cover design aspects for Phase 2 trial methods. But before we get into all that, I am just going to run through some quick bits of housekeeping first. Today's webinar is being recorded, and this recording, along with the slides that I'll be using here today, and the example files will be sent out to you shortly after we conclude. If you have any questions, please feel free to type those in the chat box, and I will do my best to leave some time for them at the end. However, if we do run into time constraints, I will be sure to answer any questions that I don't get to via email as soon as possible. The same goes for any questions that come in after the webinar has finished. I'll respond to those via email as well. You can also use the chat box to inform us if you experience any technical difficulties, and one of my colleagues will do their best to resolve them for you as quickly as possible. So before we go any further, by way of a quick introduction, my name is Dennis Desmond. I am a research statistician here at StatSols, and I will be hosting this webinar that is brought to you by Enquiry. I'd just like to start by saying thank you all for taking the time to join us today. So just a very quick rundown of the agenda for this webinar. Before we get into the first section listed here, I'm actually going to quickly go through the clinical trial process just to ensure we are all on the same page. Then I will give an overview of Phase 2 designs and discuss some of the issues faced during this phase of the clinical trial process. Next, we will focus on Phase 2a specifically, with an emphasis on Simon's design and the Bryant and Day design. We will also go through a worked example of each of those in Enquiry as well. Then we will move on to MCP-Mod as an option for combining Phase 2a and Phase 2b, and again we will look at a worked example in Enquiry here too. Finally, we will wrap things up with some discussions and conclusions, and of course, time allowing, some of the questions that may come in via the chat box as we go along. So as I mentioned, this webinar is brought to you by Enquiry, the complete solution for optimizing your clinical trial designs, from preclinical research through to post-marketing studies, with the main focus being on confirmatory Phase 3 trials, and it features over 1,000 scenarios for fixed-term, Bayesian, and adaptive studies. Enquiry is very widely used in pharmaceutical and biotechnology industries, with 88% of organizations with clinical trials approved by the FDA having an Enquiry license in 2021. Here you can also see a number of reviews from some of our customers. If you wish, you can get a free trial of Enquiry from our website, and there'll be more on that at the end, including some contact details too, if you would like to get in touch with us and get any further information. Right, so I think that we have all the housekeeping and introductory items covered, so we're going to get into the content of today's webinar, and as I mentioned, I'm firstly just going to give a brief overview of the clinical trial process. I'm sure that the majority of you are very familiar with this, but just we'll make sure to get everyone up to speed if needed. So to begin with, you have your preclinical research. At this stage, the initial research is carried out. There are various models built, be that scientific models, cell models, or animal models. And these are all used to discover candidate therapeutics. And now that we have this candidate therapeutic, be it a drug or some other therapy, we want to generate evidence that it is safe and effective. And obviously each of these phases are done sequentially. So in order to progress to Phase 2, you must first succeed at Phase 1 and so on. So Phase 1 is the first time that this therapeutic will be given to humans, though normally it's healthy volunteers at this point. And the goal of Phase 1 is to establish if this treatment is safe to administer and also the toxicity profile. So the MTD is the maximum tolerable dose, which is the dose level at which you start to see problematic toxicity events. In phase 2, then, the key question is this drug effective? And this is usually the first time that this candidate therapeutic will be given to target patients. And in this phase, we are looking for proof that this drug is successful at reducing incidence of disease, tumors, etc. And we will also look for the doses of this candidate therapeutic that will be brought forward to the phase 3 trial if successful. Phase 3 then is the pivotal clinical trial, which will be the foundation of the evidence provided to the regulator that shows that this therapeutic should be approved for release on the market. Here, there will usually need to be sufficient evidence of efficacy and safety in 2 randomized control trials or 1 large global trial. And then finally, phase 4 is conducted after a drug has gone to market. And this is a study done in real patients focusing on long-term or very rare side effects that there may be concerns about. And an example maybe that people will be familiar with is the post-marketing surveillance on the COVID-19 vaccines. So now that we've gone through that, we can move on to our first topic of the day, which is phase 2 trials. So now our candidate therapeutic has gone through all the preclinical research. It is proven to be safe, and we know those safe doses from phase 1. This is usually the first time that we will test for a clinical effect in the target population. So I say usually here because there is a growing interest in evaluating efficacy during phase 1. And one such initiative is Project Optimus from the FDA that is looking to reform dose optimization in oncology drug development. And part of that is looking into efficacy and dose selection as early as possible in the clinical trial process. Generally, phase 2 is split into 2 separate stages, phase 2a and phase 2b. Phase 2a is known as the proof of concept phase. In other words, does this drug do what we believe it does, and does it lead to better outcomes for patients? Phase 2b then is the dose finding stage. Up to this point, the dose response curve is unknown, so we want to estimate that. I should say, sorry, that the dose response curve measures the change in clinical effect as the dose changes. And we also want to use the data that we gather here to inform the doses that will be brought forward to any potential phase 3 trial. For this phase 3 trial, it will likely be a 2 or a 3-arm trial. So with that, we will need to select, the best or 2 best doses in most cases. And just to mention as well that, it's not the main focus of this phase, but throughout all steps of the clinical trial process, safety data is going to be collected. And of course, there'd be reason enough to stop if there are any concerning side effects. So phase 2 is the step of the clinical trial process with the highest failure rate, with only roughly 30% of trials advancing to phase 3 between 2006 and 2015. And this is the number for all therapeutic areas. So other therapeutic areas will have higher failure rates again, such as oncology, for example, and that is a failure rate of roughly 75%. And this also includes trials for all therapeutics, including therapeutics that replicate existing treatments like generics. So this number would likely be even lower again for new first-in-class treatments. So some of the issues that are faced in phase 2 trials. Because of these high failure rates, it's imperative that we have good designs that reject poor candidates quickly. The clinical trial process is a multi-year, multi-million-dollar endeavor. So there's no point continuing to develop a treatment that will not benefit patients. There are also ethical concerns to consider with any clinical trial. Patients taking a novel treatment are no longer receiving the standard of care treatment. So if a new treatment does not show any prospects, then we need to stop the trial as soon as possible. As I mentioned already, this is usually the first time that a candidate therapeutic will be tested in target patients. So there's large uncertainty in the dose selection at this point. Obviously, resources are finite, so there are only so many doses that we can investigate. And incorrect selection of these doses could lead to uninformative data or mean that a potentially viable therapeutic may not progress to the next phase. Excuse me, sorry. Phase 2 trials will often use a surrogate endpoint instead of the gold standard endpoints, as oftentimes these take a long time to follow up fully. And these surrogate endpoints can facilitate faster decisions. Per the FDA, a surrogate endpoint is a biomarker that doesn't measure clinical benefit itself, but is known to predict clinical benefit. And an example of this would be PFS, which is progression-free survival, or overall response rate as surrogates for overall survival in oncology studies. The problem with this, though, is you need to select an appropriate validated surrogate, and you should note that as well, high correlation is not sufficient evidence that a biomarker will be a suitable surrogate endpoint. And the relevant regulator for your jurisdiction will, in all likelihood, have a list of surrogate endpoints that have been used as primary endpoints for clinical efficacy in successful applications, that can be used as guidance here. As I mentioned earlier, there are large investment costs associated with phase 2 trials, which is further exacerbated by the fact that despite phase 2 being such a large filter, there is obviously still no guarantee of success at phase 3, and approximately 42% of all phase 3 trials fail to get approval. So due to these costs, many therapeutics will fail to progress just due to a lack of commercial viability. So now we can take a look at section 2 of the webinar, which is on phase 2a designs. Excuse me. So as we spoke about already, phase 2a is the proof of concept trial, testing for sufficient evidence of a positive clinical effect. Again, most of the time this is done using surrogate endpoints like PFS or overall survival rate. And another example is systolic and diastolic blood pressure as surrogates for major cardiovascular events. So we spoke about the issues of the high failure rate at phase 2, and this is most likely due to the fact that this is the first time that we are testing for an effect in the target population. Up to now, it was testing for toxicity in healthy volunteers, and then there was modeling done in the preclinical research, but here's the first opportunity to test this treatment in the target patients. But of course, there can also be previously unobserved side effects or insufficient efficacy as well. All this means that one-arm, two-stage trials are very popular in phase 2a. And in this type of trial, everyone will receive the treatment. And then for your first stage, you recruit the required number of subjects, measure the responses you get, and after conducting your statistical analysis, if you're below a threshold, you won't continue any further. And if you're above the threshold, then you will recruit the remaining number of subjects needed and base a decision to recommend the treatment continue to phase 2b or not on this total sample size. So essentially, stage 1 is a futility assessment. And then if you continue to stage 2, you can make a go, no go decision at this point. Excuse me, sorry. The maximum futility proportion and the minimum efficacy proportion are used to define the decision boundaries for these designs, with the maximum futility proportion telling you the proportion of successes that below which you will consider the treatment to have performed poorly. And then the minimum efficacy proportion is the fewest proportion of successes you would need to exceed in order for the treatment to be considered to have had a good response. So what are some of the methods that we can use for the Phase IIa study? So you can, of course, use a one-stage design where you recruit the required number of people and do a proportions test like a Hearn's design or a chi-square test. But this isn't as common as two-stage designs. The most popular of these would be Simon's design, but there are modifications of this like Fleming's design, which relies on a normal approximation, and Litvin's design for progression-free survival if you wish to analyze a survival endpoint instead of a proportions endpoint. There are also extensions of the two-stage design that not only model efficacy but also safety. So Bryant and Day is one very common extension of Simon's design, and Thal and Zheng is a more innovative method that models them as a bivariate distribution. There are also, extensions of these two-stage methods to facilitate three-stage designs, but these are rarer, as it will add a lot of operational complexity for not a lot of additional gain in terms of the operating characteristics compared to the corresponding two-stage design in the majority of cases. Today we don't have time to go too in-depth into Bayesian methods, but it's obviously a very popular framework, and you can see our library of previous content if you're interested. And if you'd like to know more as well, you can also make suggestions for future webinar topics. But essentially, it's a different type of inferential framework where you have a prior for what you think will happen, and then you update this prior to a posterior as things are happening. And usually these methods are done in an adaptive or a sequential way. Excuse me, sorry. Some others that I'll mention quickly: a seamless Phase 2, Phase 3 design is one in which you evaluate multiple doses in a Phase 2b type context. Say 4 doses, for example, and then you want to pick the 2 winners and drop the other 2. You can then use the data from the Phase 2 cohort to kickstart the Phase 3 trial and have a head start on recruitment there. You can also do a Phase 3 like randomized controlled trial at Phase 2, but one-arm designs would be more typical. But now what we're going to do is we're going to focus in on Simon's design, which is one of the most popular clinical trials in practice. So Simon's design is a two-stage design that allows for early stopping for futility. And with this design, you're testing a null hypothesis that you would consider to be a poor response. In other words, the proportion of responses below which the treatment underperformed relative to the expected efficacy, and you wouldn't consider to have any viability. So you're testing that versus an alternative good response that will be the proportion of responses that would lead to the treatment being seen as effective. If we skip down here for a second, the analysis is conducted using the exact binomial proportions for the responses, given the assumed poor and good outcomes. So that means you'll iteratively search over the space of all possible designs to find the one that satisfies the type 1 and type 2 errors. There are two common criteria for selecting the design. Firstly is the optimum criteria, which was the one proposed by Simon in his 1989 paper. Here, the design selected will be the one that minimizes the expected sample size. So given the proportions, it's the one that will, on average, produce the smallest sample size. The minimax criteria then selects the design that will minimize the maximum number of people who could potentially enter the study. So with this flowchart on the right here, you can kind of see a walkthrough of these steps. So you'll start off by recruiting the stage 1 cohort N1 and see how many responses there are. If it is less than or equal to R1, which is the stage 1 cutoff value, then you stop the trial early for futility. If the responses are greater than R1, then you need to recruit a further N2 subjects to reach your required total sample size. And then again, you'll see how many responses you got. If it's less than or equal to R, the final stage cutoff, then you do not recommend that you pursue this study any further. But if you exceed the responses required, then you can recommend that this treatment continue on to the next phase, which will most likely be phase 2b. And so, as I mentioned earlier, a popular extension of Simon's design is Bryant and Day's design, and it was proposed in a 1995 paper by Bryant and Day, and it's similar in structure to Simon's design, but it's a bit more adaptive, and it extends the method to also incorporate early stopping for toxicity. Toxicity here meaning an adverse event such as pain, headaches, nausea, etc. Obviously, if there are any concerns during a trial around unacceptable side effects, the trial will be stopped. But the idea with the Bryant and Day design is to have this toxicity assessment formalized as part of the study protocol. Using this design helps to tackle some of the uncertainty associated with the level of toxicity assumed, as up to this point there's only the limited phase 1 safety data available, and the number of toxic events and the severity of these in some treatments may be different for healthy volunteers, as it would be for the target population, due to the fact that some of the side effects may also be, potential or potentially caused by the disease in question itself. So, there is still despite the safety profile being found in phase 1, there's still a level of uncertainty associated with this. So this is what the Bryant and Day design helps to tackle. And again, as with Simon's design, there are two common design criteria, the optimum and the minimax. On the right then, we have a visual walkthrough of the steps. And this graphic is taken from their original paper, and it's akin to the flowchart that we had a moment ago. In the top portion here of the graph, you have the stage 1 decision tree, for want of a word, where you recruit your N1 stage 1 cohort, and then at the end of this stage, the trial can be stopped for futility, for toxicity, or for both. If, however, you exceed the stage 1 cutoff for responses, which is CR1, and the stage 1 cutoff for non-toxic responses, CT1, then the trial can continue to stage 2. And just a note on terminology there, in the paper they talk about exceeding the cutoff for non-toxic responses, but in the literature and in trial protocols, sometimes, this may be framed in the context of having, strictly fewer than some cutoff for toxic responses. So yeah, if you advance then to stage 2, you will need to recruit the required number of remaining subjects into the trial. And again, at the end of stage 2, you can say that the trial failed for futility, for toxicity, or for both. And then if on the other hand, you exceed the final stage cutoffs, CT2 and CR2, then you're in this region right up here, and you can recommend that the treatment be investigated further. And again, usually that will mean advancing to phase 2b. So now that we have gone through some of that theory, we can move on to our first worked example of the day. And here we have a real-world trial for phase 2 study for olaparib. Actually, I'm not 100% sure on the pronunciation of that. But it's a phase 2 study for a new treatment for metastatic breast cancer. And here is an excerpt from their paper outlining the study design. The main points of note for us are that they are looking to test a poor response of 5% against a good response of 20% using a one-sided significance level of 0.05. There's a target power of 80%, and under this design, the first stage sample size was 13, and there was a final stage sample size of 27. So to proceed to stage 2, there needed to be at least one response in the stage 1 cohort, and at the final analysis, the treatment would be recommended for further study if there was at least 5 responses. And just that info is summarized here in the table on the right, and I didn't mention there, sorry, but there's a minimax study design. Okay, so we now have that laid out. We can move across into inquiry. Now the general layout and functionality of inquiry has been covered in previous webinars. So in the interest of preserving time, I'll get straight into things now, but just a reminder that we have a library of previous content if you'd like to get more familiar with the software and its features. So we can start by filling in our test significance level here, just bearing in mind that it's a one-sided alpha. For the design, we'll select minimax here from the dropdown. And just to note as well, single stage is an option here too, as a matter of convenience. These designs are done in separate tables, but just if you wish to compare to the corresponding two-stage design a little bit more easier, that option is offered to you. So we'll fill in the maximum ineffective proportion of 0.05, minimum efficacy proportion of 0.2. But before I fill in the power, which is the final required cell, I'll just take a moment to talk about these two rows here. So the optimum search value is only used when the optimum design criteria has been selected. But as you can see from the help here, it is essentially the number of further sample sizes considered after a local minimum for the expected sample size has been found. So if there's a lower value for ESS in the range, then the process is repeated until a global minimum is found. And if there's no lower value found in that range, then the current sample size will be treated as the global minimum and returned as the result. So you can tweak this value here in order to increase the likelihood of finding the global minimum, and increasing it will be, at the expense of some computational time. But 10 is is usually a fairly good default value. So we're going to leave it as is. And then this lower n limit is another optional value that can be provided by the users, but it's fine to leave it blank, and nQuery will provide one. This value is used by both methods, and what it does is it basically gives users the option to specify a starting point that's still less than what you believe a realistic sample value for the sample size might be, but it's close enough. If it's not specified, nQuery will return a value that is equivalent to the minimum of the sample size for a single stage design, the corresponding single stage design, or a fixed term design for one proportion. But there's more help on all, or more info about all this in the help if required anyway. So with that, I'll just put in our value for power, and you can see we get our results here. So we get a sample size of 27 and a first stage sample size of 13, as we saw in the paper. You also get the two cutoffs as well, so the R1 of 0 and this 3 at the final stage. Now down here you have the probability of early termination, which is the probability of stopping the trial after stage 1, and then the expected sample size as well. And just with that, actually, what I'll do is I'll run this calculation again. But I'll this time use the optimum design criterion, and that way it might better illustrate some of the differences between the two. So you can see that there's an increase in the total sample size, but a decrease in the first stage sample size N1 required. There's also then a higher probability of early termination and a lower expected sample size as well. So there's no right or wrong selection here. It'll be a decision to make, keeping in mind what's best for a particular trial, depending on things like the accrual rate or the time to response, and the time to fully follow up, and all of that. The trade-off is a more decisive decision if the treatment isn't good enough versus a more measured approach to minimize the total time potentially under study. So with that now, we will move back to the slides and we can go to our second worked example. So this next example comes from a phase 2 trial for a treatment in elderly patients with mantle cell lymphoma. Sorry, I don't know the pronunciation for any of these therapies here. But again, there's a synopsis from the paper with the info summarized in the table again as well. And the key points are that they set the type 1 error for both response and toxicity at 0.05 and the type 2 error at 0.2, which is equivalent to 80% power. They fixed the probability of unacceptable complete response at 0.4 and the probability of acceptable complete response at 0.6. And then they say in an analogous manner, the cutoffs for toxicity probabilities were 0.4 unacceptable and 0.2 acceptable. So just to note here that these toxicity proportions have been translated into non-toxicity proportions in the table, as that's the terminology that was used by Bryant today, and that's what's been implemented in nQuery as well. Now, although it wasn't explicitly said, we will assume that the optimum design criteria was used. So we'll open up nQuery once more, and we'll move to this tab over here and open up the two-stage phase 2 design for response and toxicity. So we can start off by filling in our two significance levels here. And again, you have optimum and minimax, but we're going to leave it at the default of optimum. And I will just fill in 0.4, 0.6, and then for our non-toxicity portions, 0.6 and 0.8. Again, you have an optimum search value and a lower N limit. Here it's found that 4 is a pretty good default, so we're going to leave it at that, and we are going to input 80% power. Now, just with the increased complexity of this design, it's going to take a minute or so to run. So what I'll just do in the interest of time is switch over to this tab here, where I have filled in these values earlier, and we can just look at the results straight away. So again, you can see that we get the sample sizes, the final stage at 57 and the first stage at 19. So if I switch back, I don't think I mentioned that, but you can see here they have the stage 1 and final at 19 and 57. And then you also will get the first stage cutoffs for response and for non-toxicity. So you can see here that if you have 12 or fewer non-toxic responses, then you will need to stop the trial after the stage 1, and it will fail for toxicity. And again, here you have the maximum expected sample size and the minimum probability of early termination as well, again, provided. So with that, we can switch back over to the slides and we can continue on to our next part of the webinar, which is Phase IIa, IIb designs. So when doing a Phase IIb trial, you will have already established that the candidate therapeutic has some efficacy. So the goal of this trial is to estimate the dose response curve, and in doing so, facilitate the dose selection for a Phase III trial if the treatment is approved for further study. There are a large number of candidate models that can be used to estimate the dose response, with the majority of these being monotonic. So that is, when the dose increases, we also expect to see an increase in the clinical response. But it is possible to have a non-monotonic relationship, of course, but it will require more complex models to capture that shape, and we'll talk about that in a few moments. When conducting this Phase IIb trial, the model will be selected by expert opinion and or the appropriate tests, such as a contrast test, say. But what we're going to talk about shortly is MCP-Mod as a method that combines the proof of concept test with the model selection and the dose response estimation. On the right here, you can see some examples of dose response curves that were generated in inquiry using some of these possible model choices. There are 8 here, a variety of shapes, including examples of both monotonic and non-monotonic relationships. And if I just move on to this next slide here, you can see a list of these and some others. As I said, there are many potential choices, but this is a table of just some of the common ones. So the first 4 categories here are monotonic, are models for monotonic relationships, but models like beta and quadratic allow for non-monotonic relationships between the dose and the response. So that type of relationship is less common, but there are situations where an increase in dose may decrease tolerability or things like that. And so treatments with non-monotonic responses do occur in some areas, like maybe immunotherapy, for example. As I mentioned when discussing the Phase IIa methods, Bayesian methods are outside the scope of my presentation today, but I have included the Bayesian hierarchical Emacs model here, as it's an increasingly popular model in the literature especially. It modifies the Emacs model to allow for potential non-monotone curves and allows for information about the treatment effect in one dose to be effectively borrowed when estimating the treatment effect in another dose. And in brackets here, you can see the parameters required to specify the other models, but we can talk about that a bit more when we move over to inquiry. It might be a little bit clearer, so we'll keep going for now. I mentioned on the previous slide that typically the model choice is made by expert opinion and statistical tests, but at the bottom here you can see that we have MCP-Mod as a model selection procedure, and it's an approach that allows you to carry out a singular Phase IIa, IIb trial. So that's what we're going to look at now. So MCP-Mod is an innovative method for Phase II dose finding studies that's still relatively new, proposed by Bryant et al. In 2005. And it combines the processes of multiple comparisons and modeling into one single procedure, giving us the name, multiple comparisons is the MCP, and then modeling is mod, obviously. So commonly these would have been done separately, with the multiple comparisons procedure being used to establish if a dose response signal exists. Effectively, does changing the dose change the response? And to find out if there is at least one dose of potential interest. You then examine the statistical significance of the contrast tests between the doses while preserving the family-wise error rate, and you would have proof of concept if at least one dose is statistically significant. Traditional methods include a Bonferroni or similar to account for the multiple comparisons, trend tests, and Williams tests, which looks to find the minimum effective dose, or the MED, and then the MED is the lowest statistically significant dose, which is a clinically significant effect. For the modeling part, then you look to evaluate a number of subjects at each of the proposed doses, and not only to establish a signal, but then also to investigate the relationship between the dose and the response by fitting a parametric model. By doing this, you get an estimate of the responses at all doses and not just the ones directly under study. And this will give you an expanded set of candidate doses for consideration if advancement to Phase III is successful. And then down here you have some common parametric models like we saw a minute ago, linear, exponential, beta, etc. So why was MCP-Mod proposed if you can do one or both of these steps anyway? So the idea is, on its own, the MCP-Mod approach is robust without requiring restrictive assumptions about the underlying dose response curve. You're just looking to see if at least one of the doses is different from the others while controlling for the family-wise error rate. The issue, though, is that the tests are treating the doses as nominal categorical variables, so you can't really consider any doses outside of those that were part of the study. Modeling then, on the other hand, is incredibly flexible, and it examines a quantitative relationship between the dose and the response. However, the potential for problem here is that it is heavily reliant on the model chosen. So, for example, there would be very different estimates given by a linear model and an exponential model. So in 2005, when this was proposed by Bryant et al., the idea was to have a design that could do these analyses while retaining the strengths of both. There are essentially three steps. You have a design stage where you select the candidate parametric models that you wish to consider, and then you generate the optimal tests for these. At the MCP stage, you can use the appropriate contrast tests to select the best models while controlling your family-wise error rate. Just FWR shorthand here to fit in more neatly on the slide. And if you were to do a model selection comparing a number of models to the standard significance level, then that would be an issue, as you're effectively giving yourself multiple chances at finding a successful model, for want of a term. So the benefit of this approach is that you are able to do this selection in such a way as so that you don't inflate your type 1 error. And then at the modeling stage, you can fit the best model or maybe models and examine the dose response curves to find the doses of interest that can be carried forward into a phase 3 trial. And both the FDA in 2016 and the EMA CHMP in 2014 said that this methodology is appropriate for consideration, and they will accept proposals using this design. And on the right here, then you can see flowchart, which just provides a bit more detail on the individual steps and some examples of different dose response curves as well. So there are a number of advantages to using MCP-Mod. First of which, I suppose, would be the efficiency gains. There will be a cost and time saving, both for treatments that ultimately progress to phase 3, but also in rooting out treatments that fail to show sufficient promise sooner. There's also a great deal of flexibility using MCP-Mod, both in terms of the number of models and doses considered, and also, in terms of the desired endpoint, thanks to extensions to their original method, that have continued to come from further work in the area. And like a note on that then, which I suppose is also another advantage, it can be extended to multiple stages and adaptive design as well. There's lots of ongoing work in the literature with more and more design options. And of course, then there's the regulatory approval as well, which is a big advantage. But it's not without disadvantages either. It's obviously more complex and requires more expertise to implement. But we'll see in a moment that software such as Inquiry and others can help, at least with the increased computational complexity. Another issue for some might be the lack of a universal method for selecting the best model or the optimal dose. Because this isn't prescribed, it'll be dictated by the trial protocol, and so can vary from trial to trial. And then lastly, depending on the model chosen and its associated parameters, the results from some of these designs may be more difficult to interpret or explain to the layperson who might not be as familiar with this method. And there'll be also assumptions required for each of these models that you'll need to check before implementing any design. But with all that said now, we can move on to our third example, which is a work example of MCP-Mod. That comes from the 2006 paper by Pinheiro et al., in which they proposed a method for MCP-Mod, and it featured an example of a phase 2 trial for a treatment for generalized anxiety disorder. And here in the text, the relevant details from the paper itself that we need to replicate the example are stitched together. So there are 6 doses for consideration, 1 placebo and 5 active doses. It was assumed that the placebo effect was 0, that delta max, or the maximum expected treatment effect, was 0.4, and that the common standard deviation was 1. There are also 6 candidate models: linear, Emax, logistic, exponential, and 2 different beta models. You can just see those and their parameters listed here. And again, as with the other examples, there's a table of all the information summarized here as well. So they found that the appropriate sample size to achieve the target power of 80%, using the unweighted mean as the power criterion, was 62 per arm. And I'll come back to this power criterion variable in a second when we move over to inquiry. So yeah, 62 per arm for 6 active arms gives 372 as the total N. So we have the scene set here now, so we can once again jump across to inquiry, and we'll move over to the tab here for the multiple comparisons procedure modeling for continuous outcome. And as usual, we can start off by filling in our test significance level. Now, we're going to leave this at a default of 1-sided test, but obviously you can choose 2 if you wish. And we have 6 doses and also 6 models. Now you can see once I fill in these 2 values here, there are 2 side tables that will open up just below here. We will actually come back and complete those shortly, because what I'm going to talk about now is just the power criterion that was mentioned earlier. So this drop down here is needed because there are a number of ways to determine the overall level of power with this analysis. So, we're fitting a number of different candidate models. So, you can either select the mean power for all models, the minimum power, so that would be the power for the model that performed, worse essentially, and then the maximum or the median power. There's more detail on this in the Pinheiro paper, and that'll be in the references as well. But mean is a common choice because at the outset, each model is considered to be equally likely. So, it's a good default to go with. So continuing on here, we will fill in 0 for our placebo effect, 0.4 for the maximum treatment effect, and 1 for standard deviation. Now at this stage, we're going to move down to the first of the 2 side tables here, and we will put in the 5 active doses, which are 10, 25, 50, 100, and 150. And you can see that they populate the corresponding rows over here. Now, just to note that this calculation requires the placebo dose to be 0, so Enquiry has pre-specified that for you. And in this column here, you have the option to change the allocation to each dose, but we're going to leave it at the default of equal weighting for now, because that's what was done in this example. Now, in the second table here, we can put in the models and their associated parameters that we need. So I'll start off by linear Emax. This leaves us default, sorry, logistic. And we had exponential and 2 betas. Perfect. I'll just resize these actually so we can see everything at the same time. Okay, so you can see, there are brackets here detailing the parameters required for the different models. And then also Enquiry will gray out any cells, that aren't required for a particular model. So you can see, which parameters you need to provide. And then in the help here, there is a, detailed description about the different models and their parameter 1, parameter 2, and parameter 3. Just take, for example, ED50, which is required for the Emax, and that is the dose at which 50% of the max effect delta 1 occurs. But yeah, like I said, if you wish to learn more about any of the ones, there's a very useful help section provided here. So I will just fill in now the remaining parameters. Sorry, 25, 50, 10. And 85, 0.33, 2.31, 200. Okay, perfect. Sorry about that. So now that we have these 2 side tables complete, we can head back up here and enter in our desired power of 80. Now, this table relies on simulation, so it'll take, about 30 seconds or so to complete. But, just in the interest of time, I actually filled one out ahead of time again, so we can look at it straight away. So you can see here, we have our total sample size of 372. And if we look down at the side table, you can see 62 subjects per arm. If you go back up to the main table, you can see here there's the critical value for the joint multivariate T distribution that is output as part of the results. And that's this version of the calculation has implemented that distribution. And because that needs simulation, you have the fairly standard option here to set a random seed then as well. This is done in order to facilitate reproducibility, but if it's not specified, then you just get a random calculation based on your system time. Obviously, any differences in the power or the critical value will be minor as well. Like if we click back in here, you can see that it's very, very small, but the option is there should you wish. Highlighted in yellow here now in this second side table as well, you have the various model powers along with the max effect dose, the E0 and the Emax as well. I won't have time to get into it now in too much detail, but for the monotonic models, the max effect dose will be the highest dose level. But for the non-monotonic models here, which in our case is the beta models, this won't always be the case. You know, for the beta model, the max effect dose could be anywhere along the curve, and sorry, 1 second. If I click back in here, we can plot these dose response curves, and you can see that this max dose here for the beta models occurs at 100 and at 25, and not at 150 like the monotonic examples of the earlier four. But now, with that said, we will move back to our slides for one final time. And we will begin to wrap things up with some discussion and conclusions. So phase 2 trials are often the first opportunity to test if a candidate therapeutic has a positive clinical response in the target population. And it's often split into two distinct phases, 2A and 2B, with phase 2A being the proof of concept trial, determining if this efficacy signal exists for the therapeutic of interest, and then phase 2B, the dose finding trial, which establishes the doses of interest for a phase 3 study. So we saw today that good designs are crucial because phase 2 is the stage of the clinical trial process with the highest failure rate. And when you choose the appropriate design, you can facilitate faster decisions about ineffective treatments and help alleviate uncertainty around the selection of doses to be carried forward into phase 3. We saw that with multi-stage designs like Simon's design, that poor candidates can be rejected quickly at phase 2A, and with extensions of Bryant, such as Bryant and Day, I should say, sorry, that candidates can be rejected if they are unacceptably toxic too. Lastly, we just saw there a moment ago that methods like MCP-Mod can help select the best model while preserving the family-wise error rate and provide valuable insight into dose selection as well, all the while offering efficiency gains by merging phase 2A and phase 2B. So let me just keep an eye on the time here. Okay. I'm very sorry, but I'm running tight on time here, so I'm not going to get to the questions now, unfortunately. But I will be sure to respond to them via email as soon as possible. You know, my apologies for that. So if you don't have a license for inquiry and you'd like to try it out, you can go to statsols.com/trial to get a free 14-day trial with no download or install required. It can be accessed through your browser. There are no credit card requirements. You just need to provide an email address. The trial gives you access to the latest version of the software as well, and you know that will provide access to all the tiers. You know, so you'll be able to try out tables for fixed term, adaptive designs, and prediction as well. You'll also be able to take a look at the example files that will be sent out to you shortly as well. If you have any questions, you can email them to info@statsols.com, and myself or one of my colleagues will get back to you. You know, we'll just make sure to get you a response from the person that's best suited to help you out. Also, feel free to reach out to us if you have any feedback, or maybe as well, if you have an area you'd like to hear more detail on, you can suggest it and it might be featured in a future webinar. Also, there will be more information and additional content, including a library of past webinars on a wide range of topics available on our website, which is statsols.com. Quickly as well, at the end of the slides here, you will see we have a list of references. And just again to remind you that the slides, along with the recording of the webinar itself and the save files containing the examples that I used in inquiry will be sent out shortly. And that if you have any further questions, you can get in touch with us. With all that said then, that's it for today. Before we go, I'd like to once again thank you all very much for your time and also for your interest in these webinars. And I'll just remind you that we host a new webinar every month, so we hope that you will join us again soon. So yeah, thanks again, and until next time, all the best.